Welcome back to Bioanth Lab. This is David Leitner. I'm your instructor. Today I want to talk about how to actually make a Punnett square. We talked about them briefly in one of the other videos, but here I want to give you the sort of practical lesson on how to do it. You're going to need this for your lab exercises, but hopefully in doing it also makes some of the patterns of inheritance make a little bit more sense. So, without further ado, let's look at the Punnett square as essentially, it's a graphical way of interpreting the chances of a particular allele ending up in a particular zygote and then embryo. Okay, so say we have somebody who, let's just pick a random trait. We can make a make-believe trait here. Let's say on some alien planet, there are these aliens, and some of the aliens are purple, and some of the aliens are orange. Okay? So we've got, in fact, why don't I use the colors? We've got aliens that are purple, and purple is a dominant gene, and orange will be a recessive. So let's say you have one parent who is purple, but for their phenotype, but their genotype is, I don't know if that's going to be clear enough. Their genotype is mixed. They're heterozygous. So, and let's say both parents are heterozygous like this. Okay. Notice that when I'm doing this, I'm using the same, even though the colors are purple and orange, I'm just using the letter P here, and an uppercase and a lowercase. That's because the uppercase represents the dominant allele and the lowercase represents the recessive allele. Okay. I could just as easily have made the letter C for color, right? It kind of doesn't matter exactly what letter you use, but it does matter that you use the capital and the lowercase of the same letter so that you know which one is dominant and which one is recessive. Okay, so this is a representation of probability. Okay, so it's not saying that the offspring will definitely be this or that, but it says there's a particular probability that each offspring will have a particular genotype, okay? And from that, you can read the phenotype. So, in this case, to complete the Punnett square, you just connect the squares, okay? So the row and the column where things meet, you sort of bring them down. So this one is purple, purple. This one is purple, orange, right? This one is purple, orange. And notice here, I always put the dominant one first. Doesn't matter. Technically, it doesn't matter what you do, but just as a convention to make things easier to read, we always put the dominant one first. And finally, we have orange, orange. So, we end up with a ratio of genotypes of 1, right, for just purple, 2 for heterozygous, and 1 for just orange. But for the phenotypes, we end up with 1, 2, 3 purple to 1 orange. So in other words, if we were going to predict how likely is it that an offspring of this pairing will be heterozygous, we would say, oh, well, then it's 2 out of 4, right? Because there are only 2 instances of heterozygous genotypes. But if we were going to say, how likely is it that there will be a purple child, then we would look at the phenotype, and that would be 1, 2, 3 out of 4, or 3 to 1. So there's a 75% chance that an offspring will be purple, regardless of the genotype. because it doesn't matter whether it's heterozygous or homozygous for the trait. So that's the quick and easy way. This is essentially what Mendel was doing when he was starting with the second generation of his peas, right? So he would start off with true breeders. So true breeders are homozygous. So a true breeding couple would look like this, right? So each parent, remember, these are just parents here. So let's say this is, doesn't matter which, but we could say that's the female and that's the male. Doesn't really matter which way we do it. But you'll notice that when you breed two homozygous individuals, you end up with 100% of the offspring will be heterozygous, right? So, in other words, if we were looking at this, it would be 0. We don't actually talk about the 0. 4, 0, right? So there are no homozygous for either purple or orange. There are just 4 heterozygous individuals. And the chances of the phenotype being purple is 4, and the chances of it being orange are 0, right? So Mendel first started by breeding his true breeders here to get the mixed phenotype, the heterozygous phenotype, and then he bred the heterozygous phenotypes together to see what would happen. And what he notices that, you'll notice in the first generation, the orange trait is lost completely, right? Everybody's purple. But then in the next generation, the orange trait pops up again, but always in this 25%, this 1 out of 4 configuration. So that's kind of what was going on. So this is all that a Punnett square does. Now you can do more than just sort of predicting either homozygous or heterozygous parents here. You can do other combinations too. For instance, maybe you have homozygous for purple and heterozygous for the other parent. Oops, I did that wrong. See, even I can get this wrong, believe it or not. And what you end up with is, in both of these squares, you'll have homozygous individuals, right? And in this one, you'll have heterozygous, right? Because we come across and come down, come across, come down. And so here you end up with a ratio of 2 to 2 to 0. So you have 2 homozygous purple, 2 heterozygous, and 0 homozygous orange. And in terms of phenotypes, you have 4 purples completely. Okay? So that covers basic Punnett squares. In the next video, I'm going to demonstrate what a dihybrid cross is. It basically takes this same idea, but instead of looking at one trait, we look at 2 traits and how likely the 2 traits are to come together. So until then, take care of yourself, stay safe, and I will see you soon. Bye.